Good morning, everyone. As I already said, my name is Ritambhara Singh. I'm an associate professor of computer science and data science at Brown University, and I'm really excited to be here to talk about some of the integrative work that we are doing in the biomedical domain using deep learning. So the motivation behind our lab's research has always been the fact that the amount of data sets that we are collecting in the biomedical and genomics domain is growing quite rapidly. And what that has led to is the development or excitement around data-driven approaches that can effectively learn complex relationships from the data and help us develop hypotheses. And initially, machine learning was a very popular topic. And within machine learning, of course, more recently, just because we have enough volume, deep learning has become very popular and widely used. So in our lab, we basically focus on three major themes. They are integration, explainability, and application. Explainability just because of the nature of the domain that we work in, where sometimes just making accurate predictions is not sufficient. We also want to understand why the model made a certain prediction or what were the features that were relevant for the accurate predictions of the model. And we do this at multiple scales. We have a lot of work working with single cell data sets. We have also developed methods or applied methods at tissue level, so dealing with bulk genomics data set. And then more recently, we have also started looking at patient level data set, covering both the genomics data and clinical data. So in my past talks at CGSI, I've mostly focused on modeling of genomics data. Today, I'm taking a bit of a different route and going to talk about our integration efforts and the clinical side of things. So the motivation is basically the idea that now we are able to collect multiple types of measurements from the same entity. So if it's a patient, we can measure different aspects ranging all the way from gene expression to medical images to clinical information. And these data sets or modalities can be very heterogeneous. And the idea is how can we use machine learning to integrate all these different data modalities in an effective manner so that we can make accurate predictions. So in this research themes, we've had some prior work. So one of our earliest work focused on developing this deep learning framework using cross-attention to integrate imaging, genetic, and clinical data from Alzheimer's patient databases. And we showed that this cross-attention framework was way more accurate in making predictions about whether the patient had Alzheimer's or not. More recently, we have scaled this effort to multiple modalities. So as the number of modalities keep on increasing, how do we scale these large deep learning models? So we came up with this formulation known as one versus others attention, which still uses attention quite effectively, but we are able to scale more effectively and still make accurate predictions. And we have tested it on a bunch of different clinical data sets. But for today's talk, I'm going to focus on a very recent work this year we did, which was focusing on drug-based prediction tasks. So as we all know, drug development is time-intensive and costly, and there have been a lot of work done to try and make this process more efficient and more effective. In that vein, we have collected a lot of databases. So for example, one of them is known as pharmacotherapy database, which captures information or records information about drug repurposing, so the same drug being used for different diseases. We also have databases like DrugBank and DD-Inter that have information about which drugs are interacting. And this is relevant because there are a lot of drugs that can interact and cause adverse side effects. So we want to know what those drugs are and how they interact. And the idea here is that how about we use deep learning to integrate all these existing data sets and leverage all the information that we have to make accurate predictions. And those predictions could be drug disease prediction for drug repurposing, or this could be drug-drug interaction prediction to ensure about if the drugs have side effects or not. And this is not a new idea. There's a lot of work done in this space. The deep learning architecture, which is very popular in this space, is known as a graph neural network. These deep learning models are very effective at modeling graph information. And so the existing methods basically do these prediction tasks in a supervised learning manner. What I mean by that is you basically have a graph that you build using all these different databases, and they have some existing information of what interacts with what, for example, between drugs and diseases. And so you train a graph neural network on this graph representation that you have. And if you do a good job of training this graph neural network effectively, you can make predictions about new edges or new interactions based on the relationships that you have learned. And this works really well, but there are some disadvantages over here. The first one is that training these graph neural networks on very large graphs is very computationally expensive. And this is a problem because we are collecting more and more information, and these graphs are getting bigger and more richer. The other issue is that we are dealing with the supervised learning paradigm. So once we have trained a GNN on a particular task, we can't just take it out of the box and generalize it to a new task. So if we have trained a GNN on drug disease prediction, we can't just take it as is and apply it for drug-drug interaction prediction. We'll have to retrain the model. So our idea was to start looking at large language models to see how well these could do in terms of zero-shot prediction. So by large language models or LLMs, I'm talking about, say, ChatGPT, Llama by Meta, or TextSchema, which is a large language model from Google that is trained specifically on drug data sets. And the reason we were looking into LLMs is because we know that in general settings, LLMs can generalize very well to new tasks. So they have really good zero-shot prediction capabilities. So the hypothesis here is we feed in our query, like what is the interaction severity between drug X and Y, and we get a response out of it, an accurate response out of it. The other good advantage of zero-shot predictions is that it has very little additional computational cost. The model is already trained. We just apply it on a new task. So we started looking into it, and unsurprisingly, the zero-shot performance of the LLMs as is was not very good. Because as expected, the drug prediction task or drug-based prediction task we are looking at, they are very, very domain specific. So while these models have some general knowledge and some general reasoning capabilities, they don't have the information that is very, very specific to this domain. So we came up with the idea, or one of the ideas we came up with was how about we introduce this domain information using knowledge graphs that we have built out of biomedical domains. So knowledge graphs are these very rich, very large graphs that have been curated by researchers through various different databases. And what they have are these node entities. For example, they could be drug, gene, phenotype, disease. And then they have edges between them representing the relationships between these node entities. So these relationships could be these two drugs appeared in the publication together, or this drug is known to target this gene, or some drug has a side effect on something. So now you would say, well, we just talked about the computational, the expensive computation that we have to do if we are dealing with large knowledge graphs, and I'm still pitching a large knowledge graph for integrating with the LLMs. Well, the insight that we had came from a recent paper in 2021, which basically talked about this idea that while the knowledge graphs can contain thousands of nodes and millions of edges, you only need a small subgraph to make predictions for the relevant task that you have. So for example, if the task is drug A related to drug B, we don't have to look at the entire knowledge graph. We only need to look at the most relevant subgraph that would give us the answer to the task that we are dealing with. So given this idea and given this insight, we formulated our aim, which was to try and bridge the gap between the structured knowledge and the richness that we get from knowledge graphs, having the domain-specific information, and combine it with the zero-shot capabilities of large language models. Which brings me to our method. We call it K-paths for reasoning over graph paths, focusing on drug prediction tasks. This project was led by my PhD student, Ioanna, along with her co-advisor and my colleague, Carsten. But this wasn't a very easy problem to work on, so we had a big team of talented researchers contributing to this. So Ioanna, Nihal, Murtaza, all PhD students, and Steve Bach, my colleague in the CS department. So the problem formulation that we have is that of inductive reasoning. What I mean by inductive reasoning is that we have a network of observed interactions among some known entities. So this is from the database that we have. And then what we want to predict are the unobserved interactions between a known entity and an emerging entity, so an entity that we haven't seen before, or two emerging entities. So that's the inductive reasoning task. And what we are going to do is we are going to augment the information we already have from the databases using a broader biomedical network. So that's where the knowledge graph comes in, which has way more relationships between many more entities that may or may not overlap with the original network that we had. And so we call this kind of combination as the augmented knowledge graph, which is basically a combination of all the observed interactions in our database and this biomedical network that we chose. And so this creates a much richer representation for us to work with to tackle the inductive reasoning prediction task that we have. And then with this, we feed it into our method K-paths. As you can see, it has multiple steps. And I'm going to walk over each component one by one, but I just want to highlight two things over here. K-paths has actually two advantages. First is it does help with the zero-shot performance predictions of the LLM, so we can combine it to improve that performance. But if you still wanted to do supervised learning, we end up extracting these really small subgraphs so that your GNN can still train efficiently without having to deal with the computational complexity of training on an entire knowledge graph. So it has both these benefits. So let's look at the steps one by one. So first thing first, we have our query. For example, what is the possible effect of entity U with entity V? Next, we have our augmented graph, which is the external graph that we have, knowledge graph, plus the network of observed interactions. And from this augmented graph, we basically extract K reasoning paths between our entity U and V. So these are K shortest paths between two entities using something known as the Yen's algorithm, and I'm going to describe that algorithm in a bit. What we get from this algorithm are a diverse set of reasoning paths between the two entities, but there are some relational redundancies that we also get. So we apply some sort of a diversity filtering that filters out any sort of relational redundancy that we got. And then that gives us our final diverse set of paths. And from those, if we are working with an LLM, we will textualize these paths into natural language so that it can be fed as descriptions into our LLM inference. And so we feed it with the query and we get our output. However, if we wanted to still do the supervised training task, we can basically convert these diverse paths into much smaller subgraphs that allows us to do a lighter GNN training and still give good results. And finally, talking about the Yen's algorithm. So this algorithm is a way to find the K shortest loopless paths, and it goes a little bit iteratively. So the idea here is that given my subgraph and my two entities U and V, Basically, I'm going to use any shortest path finding algorithm, Dijkstra, breadth first search, depending on whether you have a weighted or unweighted graph. And so I get the shortest path between the two entities. So that's my k equal to 1 path. There is another path technically between u and v, but it's not the shortest path, so we are going to disregard that and keep the shortest path. So that's k equal to 1th path. Now we want to find other possible paths. So what we do is we are going to remove the edge that we had in the first path and then look at the alternative shortest path. So that gives us k equal to 2. Similarly, we'll remove the 2 other edges and then whatever remaining shortest path we can find between the two entities. So this is how we get multiple k paths. And like I said, there can be some relational redundancy which we filter out as an additional step. So now that I have explained the method, let's look into the experimental setup. So the knowledge graph that we are using is a very popular one in the field called HeteoNet, which has been curated from 29 different databases. It has entities like compounds, genes, diseases, etc. Once we had processed the HeteoNet, we ended up with around 33,000 nodes and 1 million edges and 23 different relation types. In terms of prediction tasks that we tested our method on, we picked the drug disease prediction task. So from the pharmacotherapy database, this has 3 prediction categories talking about, X, does drug X interact with drug Y, disease Y, and so on and so forth. The other interaction task that we focused on is drug-drug interaction prediction. So here we have a DD-Inter database that has, again, 3 categories of predictions talking about how severe the interaction between the drugs is. And then we also included the drug bank database in this task, which has 89 different possible predictions capturing the metabolic interaction level. So this is a little bit more comprehensive. It has things like this decreases the rate of that. So as you can tell, I have basically sorted these tasks going from the simplest to the most complex, with the drug bank related being the most complex prediction task. In terms of baseline methods, for the evaluation of the zero-shot performance, we basically picked large language model-based baselines. We had 2 settings for them. One was just the very base setting, which we tested very early on, which is if we just feed in the query, how much information we can get from the general knowledge that the model has. And then the other setting was we also added definitions of the 2 entities from which we were trying to make the prediction. So if there were 2 drugs, we added the definitions of these drugs as additional query input. For the supervised task, we basically wanted to see what is the computational efficiency of training on our subgraphs. So we compared it with the state-of-the-art GNN models trained on the complete knowledge graph that we have. So first, let's look at the results for the zero-shot setting. So can we predict interaction types, severities, indications reasonably well in a zero-shot setting by reasoning over our k paths? We tested it on multiple large language models, but for this presentation, I'm just picking 2. Llama, just your general LLM. TextSchema is also an LLM, but it has been trained on drug-based datasets. So when we look at the drug disease prediction task, we see an improvement in performance when we use k-paths, though the performance improvement is more drastic for your regular LLM and not so much for TextSchema, which I think is expected. When we start looking at drug-drug interaction prediction task, we see more improvement in while using k-paths for both the models. And then when we look at the most complex task, which is 89 different predictions for the drug bank data set, we see a drastic improvement in the performance. I don't even have to highlight it. And what I would like to note here is that none of these models have been trained on any specific data set or any specific task. This is just augmentation generalizing in zero-shot setting across the 3 tasks. And because we extract these very easy to understand reasoning paths that we feed into the query, we can also kind of see why the model is making whatever predictions it's making. So to give you an example, suppose this query from DD-Inter, what is the interaction severity between these two drugs? The LLM only model says moderate, which is incorrect. The LLM with definitions also says moderate, which is incorrect. But with K-PATS, the model gives the correct performance. And so if you look at the reasoning path, you can see why the model was able to make an accurate prediction, because we have provided it some information about the genes that the two drugs bind to and the side effects the two drugs have individually. So combining them would lead to a major side effect. Another example from pharmacotherapy, what is the effect of this particular drug on muscle cancer? LLM only model doesn't give the right answer, neither do the one with definitions. But then when we add K-PATS, it gives the correct answer that this is disease modifying, again because the reasoning path suggests that the drug treats kidney cancer, which is related to muscle cancer, and it down-regulates this particular gene, which is also associated or related to muscle cancer, so it's able to make accurate predictions there. Lastly, we also test the usefulness of this K-PATS algorithm for supervised settings. So like I said, what we are going to do is we are going to take these state-of-the-art GNN methods, MRGNN and relational GCN, and we are going to train it on the complete graph versus the subgraph that we extract using K-PATS just to see how well we do in performance. And what we find is that the performance, there is barely any difference between using the full graph and the K-PATS, but there is a significant difference in the amount of data that we are working with. So with complete graph, we are looking at some 1.7 million triplets, whereas with K-PATS, we have reduced it to just 100,000 triplets, which is way less. Similar results for drug-drug interaction prediction as well. The performance remains roughly similar to using the complete knowledge graph, but the big difference is that the knowledge graph that the model is working with is reduced almost 90% by size, so thus improving the training efficiency. So to summarize, K-PATS is a novel path retrieval framework that extracts relevant and diverse reasoning paths from large biomedical knowledge graphs. It enables LLMs to effectively make predictions on unobserved interactions between entities in a zero-shot setting. If you want to use it for supervised training, that's fine. It reduces the computational burdens of GNNs. And because you can see these reasoning paths, you can also kind of get some insights as to why the model is making certain predictions. The paper is available as a preprint, and the code is available on GitHub, and this work will be presented in KDD very soon, actually. And last but not the least, I would like to acknowledge all the people who worked on this particular project. So they're all in bold: Amina, Ioanna, Murtaza, Carsten, Nihal, and Steve. I would also like to highlight the two people I have underlined. So Amina and Michal are both final year PhD students who have worked on very interesting method development for data integration in the biomedical domain, and they will be on the job market in the fall. Finally, a big thanks to my lab. As you can see, we are a happy group of students and me, and we are very interested in solving these interesting biological and biomedical data integration challenges using machine learning. Big thanks to our major funding sources and my affiliations, and I'm happy to take questions now.